Okay, it's couple minutes after 8. I think we will start, and people will keep trickling in as we go. Thank you all for joining us today, and happy Leap Day. We have a very exciting talk. I hope you can hear me. Can you hear me?
Yes.
Yep.
Yes.
Okay, thank you. Yeah, so we have a very exciting talk by Dr. James Fulcher, but before we go into that, let me just thank to all of our annual sponsors once again that are supporting the work of the consortium and the entire top-down community. And remind everybody about a number of different resources you can reach at the consortium site. We have had a number of very exciting and entertaining speakers so far, and we would like to thank them once again. Our next speaker is going to be Jake Melby mid-March, and I hope you all will join us for that chapter in this book. Today, coming back to James, I would just like to briefly introduce James. I've been working with James over the past couple of years quite closely. He comes to us after obtaining a PhD at the University of Utah, where he was doing mostly organic synthesis, making these large peptides and proteins. And then when he joined PNNL as a postdoc, he primarily worked in the area of top-down proteomics as applied to different diseases, particularly brain-related diseases and diabetes. And then most recently, he also became interested in single cell and spatial proteomics and is actually leading our work in developing technologies for parallel measurements of different omes from the same sample, same limited sample, such as single cell. Today, he will tell us about some of the work he has done to enhance quantitative top-down proteomics. So I think he has about 30 minutes to go through a couple of slides, and then we can afterwards talk about any topics that you're interested in, or he will be happy to answer any questions that pop up. And with that, James, the podium is yours.
All right, thank you, Lily. Let me see. All right.
We can see your notes. Just go to the display settings on the top and change it. Swap.
Yeah, okay, there we go.
Thanks.
Yep.
Go ahead.
Yeah, thank you for the introduction, and thank you to the organizers for inviting me to present on my work in top-down. So as Lily mentioned, I came from the University of Utah with a PhD in biochemistry, and I worked in peptide and synthetic peptide and protein synthesis, but I knew I wanted to learn proteomics, and when I was looking for postdocs, I saw this opportunity to do top-down proteomics, but boy, did I not know what I was getting myself into. Regardless, it has been an enjoyable experience, and I'm really happy to be able to present the work that we've done over the past few years. So, when I first got started in top-down, I wasn't sure exactly how to process the data, and at the time there were so many tools, and a lot of these tools I've listed here. I want to just briefly mention. There's so many different avenues for analyzing top-down data in different ways we look at it, and I've used all these tools to some extent in different areas, and I think they've really served some great purposes. And even the more recent developments as we move into quantitative top-down proteomics, I think that there's a lot of room for growth as well for these tools. And so when I first started, I was looking at through these tools, and one that stood out to me was TopPIC, and as I'll discuss, there were some advantages to using that software that I felt really lended it to doing the type of analysis that we were trying to do on a large scale. So many samples of and trying to do quantitative top-down proteomics across a large cohort. And so TopPIC was interesting to me or useful for us for a few reasons. One, it could be run in parallel. So that what I mean is that I didn't the software itself was not locked to a single computer or licensed to a single computer. We could run it on a cluster if we wanted. We have the ability to identify open modifications, so unknown modifications through an open modification search. And what I observed is that while this was pretty powerful, we could get more identifications. The interpretation of these open modifications was somewhat of a challenge as well. And FAMES was very new when I started. One of my earliest works was implementing FAMES and top-down and finding that it works. It really does help, I think, increase the number of proteoforms we can observe. And early on, TopPIC was, at least to some extent, was FAMES compatible with a few workarounds. But more importantly, in the output of the results of that we get from the tool, the MS1 and the MS2 data that we get also contain chromatographic information. That was important for some of the concepts that I think or some of the things we wanted to do with quantitative top-down proteomics. So there are a number of challenges when we consider how do we do robust quantification of top-down, and think about kind of what how it's been done in bottom-up proteomics. And so a lot of the tools that exist in TopPIC, is somewhat like this. So if you think about, let's say you have 200 files or 200 data sets that you want to analyze. You can run these on a single computer with a single instance of TopPIC, but on average, one analysis of one of our data sets is 8 hours. And so if we have 200 data sets times 8 hours, it's 2 months of analysis time on a single computer. But the advantage is we get one output file. All of our data is more or less concatenated, and so there's consistency with the proteoform identifications. Now, there's also the ability to do parallel analysis, where we take these data sets and we divide them across a cluster of computers. And say like we had 30 computers. This gets us down to 2 days of analysis time. But the problem is that now we have multiple outputs, and we have to figure out how to what proteoform A in one of these outputs, how do we correspond to the proteoform A in the other outputs? So this is actually the approach here at PNNL that we often use, or that we have the capability to do. And this kind of spurred some of the developments of why we needed to improve on what we could do with TopPIC output. As I mentioned, FAMES was compatible with TopPIC, but when we have these fractions, and so I'm showing here actually 2 papers that related to the on the publication of FAMES and top-down proteomics. But one of the issues that comes up is that a single proteoform could be observed across multiple compensation voltages. So a compensation voltage is more or less a gas phase fraction of our sample that we're analyzing. And some of the challenges are, well, when we want to do quantification, we want to make sure that we're not, that in one of our data sets, maybe we only see proteoform A in a compensation voltage of -40, and then it's -30 in the other. And so that it kind of confounds the quantitative aspect of what we're trying to do. And so there are special considerations there that we need to take into account. Proteoform ambiguity, this is a big one. Open modifications are a great example. It's very challenging to know across data sets if the same proteoform is being reflected. And sometimes what can happen is that even across a single peak, if you sample it multiple times with an MS2, an MS2 event, you can get different proteoforms from the same peak just based on some of the stochasticity of the MS2 fragmentation, or selecting a different charge state of the same proteoform can give you sometimes different results. Match between runs has been, I think, a very important or powerful approach in bottom-up proteomics, in particular for increasing sensitivity and just reducing the amount of missing data we have to deal with. But implementing this in top-down proteomics requires some new developments as well. And finally, visualization. So one of the things that I really was surprised about when I first got it into top-down was just how many fragments of proteins there are. And this is, of course, I think this crowd recognizes that the instruments we use kind of constrain us to seeing proteins under 30, 35 kilodalton, depending on how, what type of analysis we're doing. But when we do these kind of denaturing top-down experiments, we see so many of these fragments, and so it kind of raised in my mind, like, well, what is the importance of these, and how do we interpret them in an easy way? So a lot of those challenges kind of came to into the development of TopPIC-R. And so TopPIC-R is an extension that's implemented in R of the TopPIC outputs. And so this flowchart, kind of describes all of the different steps that are involved. This work was performed with Vlad Pechik and Evan Martin and myself as well. And we set out to address a lot of those challenges. And we implemented, I think, some approaches that have been already utilized in bottom-up proteomics and that can make for robust quantification. So I'm going to highlight really only two aspects of TopPIC-R in this talk in the interest of time. One is parsimonious inference and proteoform level assignment. So this is very important for understanding how much ambiguity exists in the identification that we're looking at, because we can have, the output just tells us that what the proteoform is, the sequence, but how many genes does that that proteoform map to, or is the PTM localized? Is it actually is this unknown modification? It doesn't represent a single PTM or multiple PTMs. So there's a lot of ambiguity that can exist in a proteoform identification. And then the clustering aspect, I think, is really important. So being able to distill down, to go past the proteoform identification and say, wait, this proteoform in one data set, we're calling one thing, and then in this other data set, we're calling it something else. But really, when you look at retention time and the M/Z or mass domain, there's an underlying feature that is basically the same for both. And so that's where clustering comes, or clustering and also the retention time, alignment, and mass calibration steps are important for being able to do this match between runs and say what is a proteoform across many data sets. And so we demonstrated this this tool in a test data set from a prior publication, the Informed Proteomics pipeline. So this is 10 human and mouse xenograft breast cancer samples. So to start with thinking about parsimonious inference, let's take an example. We have our TopPIC search output. We have proteoform X. Proteoform X can be mapped to 4 different genes, one of which is in SwissProt, and 3 of which come from Trimble. And so we often use in our analyses the Trimble database because we do find that there are many identifications that pop out in the top-down data that we don't see in bottom-up. But we have the situation where we have ambiguity between A, B, and C, and D, right? Now, let's say that if we look at the number of uniquely proteoforms that can be uniquely identified to these different genes, we see that gene A has 1, B and C have no. Four. And so this parsimonious inference is kind of an assumption of, well, the gene D has the largest number of uniquely identifying proteoforms, and perhaps it's the most abundant. And so what we do is we take proteoform X and we say, okay, proteoform X is probably derived from gene D. So this is kind of a way of saying, like, most likely it seems that gene D is the origin of proteoform X. Now, of course, we reflect the ambiguity with the proteoform level. So level 2D tells us that that particular proteoform, assuming there's no PTMs or any other aspects of it that could involve more ambiguity. But 2D reflects that we're certain in the PTMs or the localization, the sequence, but we are uncertain in the gene of origin. But gene D, so what we do with TopPIC is we make an assumption about what seems to be most likely, and that would be gene D. So there was a prior publication that showed, that TopPIC could not necessarily assign all the proteoform levels. The output doesn't include all the appropriate information to do that. And so we were interested, of course, in implementing, the ability to identify proteoform level in the TopPIC output. And so TopPIC-R does have the capability. We can do that with some of the functions that we've built into the that R package. And so that test data set I referred to, we can see all the proteoform levels that we could assign. So from, a level 1, which would be no ambiguity, down to a 4B or 3E, which represents multiple levels of ambiguity. So when we think about clustering and match between runs, to me, this is the more important for the quantitative aspect. We have, in this particular example, which I took from our TopPIC-R paper, we have we're basically plotting every proteoform spectrum match across the 10 data sets. And this is for one particular gene, which I can't recall, but in the paper, this is one gene that has many different proteoforms. And so you can see that each color represents more or less a cluster that we've identified through hierarchical clustering, and the cut height is determined through the determination of how much mass error there is and how much retention time error there is. And if we zoom in and we look at, say this mass range and this retention time range, we can see, the clusters that are showing up in here. And so this is helpful for coalescing, I think, or distilling down to the feature. And this is important because when we do match between runs, we're going to go back to the MS1 feature file, which has all of our unidentified proteoforms. And we're going to look for a particular mass and retention time. And so we have to perform the retention time alignment and the mass calibration, which we apply, and we develop a model based on our data that says, this is the appropriate way to correct for those variables, and we apply it to the MS1 features. And then we'll take the tolerances we determine in terms of the error for retention time and mass and look in the feature files to see if anything fits within those criteria. And so when we zoom in and we look at these clusters here, there's two also interesting examples that I think often comes up in top-down data. And this is the isotope log cluster group. So often identifying the monoisotopic mass is a challenging thing in deconvolution. And it's not uncommon that, the experimental mass that we see is 1, 2, 3, or 4 Daltons off, in particular with open modifications. And so TopPIC-R looks at the clusters we first identified and then looks to see if there's any isotopologs that are within a close retention time or mass distance of 1 or 2 or 3 Daltons. These are user settable parameters in the function in R. And then we would group this into a single proteoform cluster. And so this really improves the reproducibility of our proteoform IDs across data sets, in addition to being able to do match between runs once we have this information. And so just applying match between runs to the 10 data sets could reduce the proportion of missing values from 54% down to around 23%, which was a significant improvement. As I mentioned, one of the challenges are visualizations, and fragments of proteins are so often observed in terms of what we're seeing in top-down data. And so we developed this kind of proteoform mapping visualization that tells us a lot of different information. So this particular example is GAPTH, and we have many different fragments presented here. So the starting amino acid is, or the amino acid position of the total protein is shown at the bottom, and we have the starting and ending positions of all the fragments that are being mapped here, and the color is indicating the spectral counts, the abundance roughly. And we also can map modifications. So the PTMs that are identified by TopPIC, excuse me, still getting over COVID from a few weeks ago. The PTMs that are identified by TopPIC, we can map onto these proteoforms. And even the unknown modifications, we actually have the ability to, we import the Unibot database that into TopPIC-R, and we can actually search the unknown modifications against that database to see if there's anything that closely matches within a PPM tolerance set by the user. And so this is very helpful for, I think, looking at across the proteoforms and starting to see patterns. I mean, not just the, where there's favorite cleavage sites that we can see, but also PTMs that are being localized to, say, a very specific position across different proteoforms. Like in this case, this disulfide is at a particular cysteine on many different proteoforms across GAPDH. So it implies that there's potentially something, important, or at least very or high enough abundance that we reproducibly observe it across many different proteoforms. It's easy to see with these visualizations. So that is kind of the, I think, big view of what TopPICR was about, and I'll show a few examples of how we applied this. But in terms of dealing with the separate output files, TopPICR has functions for concatenation of all the files and being able to apply global FDR at the protein or isoform level. And some of the FDR, I should mention, our FDR approach is very similar to, I think, what's been published by Kelleher and colleagues on some of the FDR approaches that we should take in top-down. The FAMES fractions, something I didn't mention is that when we have a proteoform that's observed across multiple CVs, another question is, well, how do we roll that up to, or how do we bring that to a single value, an abundance value? And so fortunately, there's roll-up algorithms that have been developed for bottom-up proteomics for rolling up peptides to protein level. And so similarly, the same approach can be taken with proteoforms observed in multiple FAMES gas phase fractions. There's also the proteoform ambiguity. As I mentioned, we showed that we can identify proteoform levels from 1 to 5, and then clustering also helps to reduce this ambiguity. And so when we actually identify a cluster, oftentimes there are many different proteoforms that are have been identified for the same feature. And so we can actually look at all the identifications across a number of data sets and actually pick out, based on the abundance or the number of spectral counts, which one is most representative of our proteoform cluster. So it's kind of a putative proteoform. Match between runs, and as I demonstrate, with retention time alignment and mass calibration, so having the chromatographic information really helps us to be able to do a robust match between runs, and this significantly improves the data, the proportion of missing data. And so, in terms of when I think back about what we've done, so there's so many different applications of top-down that I've been very fortunate to be a part of here at PNNL. And I think TopPICR without TopPICR, it would have been very challenging to be able to do this. And so each of these studies I've highlighted here have in some way involved TopPIC and TopPICR. And so I think where I really want to highlight in the context of this talk are 3 particular studies. So one is involving mouse islets with a specific enzyme knockout. We have the soybean root nodules of top-down that we've applied top-down proteomics to, and then probably what has driven most of the development of TopPICR is the large-scale investigation in Alzheimer's disease. But we've also applied it in the context of HubMap and using it with matching our identifications to MALDI MSI data of proteoforms from tissues. Okay, so insulin processing obviously plays a role in type 1 and type 2 diabetes. And so Bruce Fulcher from UBC had an interest in looking at carboxypeptidase E and its role in the processing of insulin in the context of diabetes. And so they have a beta cell specific CPE knockout mouse that they were using in their lab. And CPE has a very particular role in insulin. So insulin, as it's being processed, there's initial cleavages by proenzyme convertases 1, 3, and 2. And then CPE will come in and trim off these basic amino acids. And so I think what you can appreciate is that if we knock out CPE, there's an expectation that those basic residues will presumably be left on and will be changing in abundance. So this is very much a proteoform, requires a proteoform approach and thinking about how do we quantify these. And so Bruce and Wei-Jun Khan from PNNL came up with this idea of like let's apply top-down proteomics to this. And so the basic workflow that we applied was to have 100 islets from wild type or beta cell CPE knockout mice, and to utilize the FAMES method, which I referred to, with internal CV stepping, and more or less really leverage TopPIC-R to do quantitative top-down proteomics here. And so what we found was that we could quantify a fairly large number of proteoforms, 1,400, and insulin, because insulin is such a high abundant protein within beta cells in these mice. The vast majority of proteoforms, or a very large proportion of the proteoforms, came from insulin. And so when we look at the structure of insulin, we have the A chain, the B chain, the C peptide. And these red residues represent residues that are meant to be cleaved by carboxypeptidase E. Now, when we knock out carboxypeptidase E, we would expect, of course, these to be left on there. And that's more or less exactly what we see. So if you look at this volcano plot here, these colors actually correspond to this A chain, B chain, C peptide, or dibasic residue containing. And so each dot is a proteoform that we quantified. And the B chain proteoforms represent proteoforms that could have PTMs, that maybe they are truncations, but each blue dot represents a proteoform that has this sequence on here. And then any proteoform that contains one or multiple of these basic residues is colored in red. And so when we knock out CPE, we see a lot of these red dots are higher in abundance, and that's fortunately exactly what we would expect. And similarly, or maybe even more interestingly, a lot of the A chain, B chain, C peptide proteoforms are decreasing in abundance. So this reflects not just changes in the composition of the proteoforms, but also the turnover. So things that because of disrupting these upstream processing steps, the downstream steps, the degradation or the turnover of the insulin has been changed in some way, such that we're seeing less of those fragments. And so another way to look at this is to use that same visualization I showed before, but to map the log2-fold change in abundance, excuse me, across these different proteoforms. And so you can actually see, I think even more clearly here, that if I've pointed out to these canonical B chain, proteoform, or the A chain, and so overall we see that insulin is decreased, but we can also see the increase in the dibasic residue containing proteoforms. And so anything that is, you see these dark red, it means that they've gone up significantly in abundance. And if you look at where the dibasic residues are along the intact protein sequence, you can just draw your eye and look up and see that when proteoforms contain residues in this area, they are most often being increased in abundance, with some exceptions that might be related to turnover. Now, the study not just was, we didn't just look at insulin itself. There are many different hormones processed by CPE, and we could define substrates of CPE that were both canonical, basic residue containing, but also some non-canonical, even histone proteoforms, which have basic residues on their termini, appeared to be cleaved by CPE. And it's interesting because I think it really tells to kind of the impacts that can happen when you remove such a critical enzyme. We showed that the up and downstream hormone processing could be impacted by the CPE knockout, and this that there are also compensatory cleavages by homologous enzymes, right? And so the mice that have the CPE knockout, for the most part, are fairly normal. They just have somewhat of a diabetic type phenotype, being overweight and having insulin resistance type issues. And so what we found is, of course, that even with the loss of CPE, there's homologous enzymes such as carboxypeptidase D that may be involved in ensuring that insulin gets produced appropriately. And so, this was, I think, an excellent study that really benefited from being able to have TopPIC-R. All right, so moving into soybean root nodules, this is kind of transitioning from to a completely different background, but top-down proteomics can be applied really to anything. And I think this study was pretty interesting in itself. So Gary Stacy he's a big player in the field of soybean or plant biology, and so he has worked a lot on soybeans, and in particular soybean root nodules, which have these bradyrhizobium bacteria that are nitrogen fixing. And so what I've tried to portray here is that if we have a root that from a soybean, there's these root nodules in red, we have these bacteria within them that are actually constrained within the plant and producing or nitrogen fixing and producing molecules that the soybean takes advantage of. And so Gary had a nitrogenase knockout bradyrhizobium strain that we could compare to a wild type. And so we wanted to compare root nodules that had nitrogen fixing ability and nitrogen fixing deficiency. And so we wondered, how is the intact proteome impacted by this nitrogen fixing deficiency? And this work was performed with Lily, Moe, and Kevin from all from PNNL at that time. And what we could see with top-down proteomics was pretty interesting. So there was a very large number of changes in both organisms. I mean, naturally, this is a very disruptive type of perturbation. But across the 1,600 quantifiable proteoforms, 738 were from soybean and 910 were from the bacteria. And we can see that there are a number of changes in both directions in terms of and having nitrogenase deficiency and the wild type. And we found a number of proteoforms that I've pointed out to some extent here that had very unique modifications. But perhaps what was very interesting was leg hemoglobin. So leg hemoglobin is a very high abundant soybean proteoform. And the main function of leg hemoglobin is to facilitate the oxygen supply to the nitrogen fixing bacteria. Now, leg hemoglobin is produced by 4 different leg hemoglobin genes, LGB1, 2, 3, and A. And this volcano plot is showing the leg hemoglobin proteoforms from these 4 different genes and the characterization of some that are truncated and some that are full length. And so what's interesting is that when we have nitrogenase deficient root nodules, full-length leg hemoglobin is more abundant. So that's what we're showing here in purple, that more of these full-length leg hemoglobin proteoforms are found. But then in the wild type, we see many more truncated leg hemoglobin. And so what's really cool about this is that, once again, I think when we think about these fragments, we're starting to kind of get an idea that, okay, there's something changing with the turnover. And what's even more interesting, I think, is that, we also did bottom-up proteomics on these. And so it's very easy to do bottom-up, get thousands of proteins and to make conclusions on that. But what's very interesting is that none of the leg hemoglobin genes are changing in absolute abundance. But what's clear is that the turnover has changed considerably. And so what we think this reflects, of course, is that there's reduced metabolism probably in the having fewer of these nitrogen-fixing bacteria or bacteria that are deficient in nitrogenase activity. Have less metabolism, and so they perhaps what happens is that leg hemoglobin may be chewed up into pieces once it is transferred to the bacteria, just because of the nature of the nitrogenase activity. And so it's pretty interesting. I think that TopPIC-R once again really helped us to do the quantitative aspect of this that pulled out these interesting results. Now, as I mentioned, we developed TopPIC-R. And what I'll say is it with in mind trying to do a large scale study. And so Vlad has been the lead on this since I started as a postdoc. And we've been working with Ashley from the Kelleher lab recently, who's now at PNNL, and then Phil DeJager and Dave Bennett from Columbia Medical University or Columbia Medical School and Rush Medical University. So they have an interest in Alzheimer's, and so we have access to brain cortex samples from the Ross map cohort. And this enables the interest we had was doing top-down in this context of Alzheimer's. Now, this required a lot of, I think, developments on and the method development, not just for data analysis, but also for just processing the samples. Because, you know, if you're familiar with Alzheimer's, amyloid beta is one of the main proteins of interest, and it's very hydrophobic. It's challenging to isolate. And so we came up with a very interesting differential solubilization technique that produced two fractions. Now, that's in addition to the fact that we have these famous gas phase fractions. So if you think about, you know, what ends up happening, we basically take 103 brain samples, multiply it by 2 for these differential solubilization fractions, and then multiply times 3 for the gas phase fraction. So this data ends up complex very quickly, and it was actually over 3 terabytes of data from 206 runs, and this gives us 618 TopPIC output files, and actually I think it's more around 1800 because you have to multiply by 3 for each FAME CV. So this was a very, I think, complicated study, but TopPICR really helped to make it possible to do what we so often do with bottom-up proteomics. And so when we looked at just overall the big picture, I'm not going to talk too much in this talk about the biological findings relevant to Alzheimer's, but I'm going to talk about what TopPICR enabled us to do. And so there are over 11,782 unique proteoforms we found in our study. On average, per individual, we observed 6,250 proteoforms derived from around 856 proteins. And so the scale here is very large. And then when we consider, after using TopPICR and having a match between runs, we could have over 6,000 proteoforms that have fewer than 50% missing values, which is important for the quantitative aspect of what we want to do. And of course, related to Alzheimer's disease, we pulled out a large number of amyloid beta proteoforms that we could quantify. As an example, and so if you think about the context of Alzheimer's disease, there's this protein called MAPT or tau. So tau is the kind of another bad guy in the Alzheimer's disease pathology. And so we see many proteoform spectrum matches for this, but there's an importance here of the proteoform level and knowing the ambiguity. We have over 2,000 spectral matches to tau across, I think, probably 7 or 8 splice isoforms. So this is a challenging aspect to decide, okay, which of these fragments or, you know, which of these proteoforms belong to which tau variant. And what was interesting is that, you know, we have over 1,000 that can be assigned to 3 different splice isoforms presented here. And so these N1 and N1, N2 domains, R1, these are different repeats that can be present on the different splice isoforms. And there's the N1 and N2 really differentiate some of these spectral matches. So we have 930 that are corresponding to this 1N and 4R or 1N and 3R splice variants. But we had 86 that were unique to one particular Trimble ID, which had a 41 amino acid exon insertion between the R2 and R3 repeats. And so, you know, without TopPICR and having the ability to assign the proteoform level, we wouldn't have been able to really tease out, is this completely, is there ambiguity here? And actually, these are very unambiguous. So it really helps to show that, you know, we have at least 86 special matches that show that this very unique splice isoform, which really, you know, only exists computationally, is probably being expressed in brain tissue. Thinking about the retention time alignment, the clustering. So there's another example here that I think is very unique and really couldn't have been done without those steps in TopPICR. So haptoglobin has a few alleles, and 2 in particular here are HP star 1F and HP star 1S. The fast and slow actually refers to its oligomerization capability. And so these 2 alleles are characterized by a very unique change. And so it's actually 2 amino acids in asparagine and glutamic acid that can be changed to aspartic acid and lysine. And so you might think that a change like that would induce quite a we should be able to easily differentiate these, but actually they're isomers. There's no change in the mass at all. However, there is a very strong change in the retention time. And so what this figure is showing is that we have the two alleles. They're more or less the exact same mass across these proteoforms, right? And so the mass is on the y-axis. But the two different alleles separate out by retention time. And so being able to do the retention time alignment in TopPIC-R was, I think, very important for being able to separate these clusters and say, okay, these are actually two different alleles, because the MS2 data wouldn't tell us anything different between them. And it would be very actually, if you include both of these in the database, you'll just get identifications for each of these within these clusters. So I think considering retention time and having the capabilities we have in TopPIC-R really help to reveal unique nucleotide variants. And so, as I mentioned, I'm not going to talk too much about the biological aspects of the Alzheimer's data, but I think there's some concepts here that I want to talk about that I feel, I hope that if you take away anything, it's kind of thinking about, well, how do we get new views of our top-down data? And so I've had an interest in implementing, or at least integrating, structural data with these fragmented proteoforms, because it just seems that there are certain, when you look at the proteins and the way they're cleaved and they're broken apart, there's very unique patterns that are very different and defined for each protein. And so I wondered if there was some structural aspect that might be contributing. And so if you're not familiar with AlphaFold structures, I'm showing here for a protein MAP1B. And these red and or yellow and orange areas out here, these squiggly lines are very low confidence scores. And so that's what these colors represent. Blue scores represent high confidence AlphaFold prediction scores, and orange and yellow are low confidence. And so this MAP1B is a very interesting example. And I think what we can show is that we can integrate this data into our TopPIC-R visualization to start getting a better idea about how these proteins are processed. All right, so this figure here, we have different fragments or proteoforms that we identified across our 103 subjects, and they're being mapped to the position along MAP1B. Each of these colors down here represent the model confidence scores from AlphaFold, and we've mapped them to each amino acid. And so all this red and orange area also corresponds down here to this green area, which is these are annotations from UniProt, which we can map here, that have been identified as being disordered, presumably experimentally or computationally. And so what's interesting is that across, I think maybe there's 100 or so proteoforms here. They're all being cleaved within the disordered region. And so this, I think this really highlights just an example of there's some interesting stuff going on here in the way that these proteoforms are processed, the way these fragments are being derived. And so this is the kind of a thought experiment and implementation that I think can be useful for many experiments. We also see something very unique regarding SH3 domains. And so once again, we have a few fragments that I mapped here for this protein SH3GL1. So this red area is an SH3 domain that's annotated by UniProt, and then we have a disordered region that's also annotated here in this orange. And what was interesting is that this cleavage was being found, was very specific for this orange disordered region, and seemed to contain the entire SH3 domain. Now, this might not be a unique observation in itself, but it's very consistent across many different SH3 domain containing proteins. I've only highlighted 6 because that's what could fit on the slide, but there's actually many more than this that we find in our data where the C-terminus, there's C-terminal fragments that have the entire SH3 domain separated from the intact protein. Now, of course, identifying the intact protein on these, that's a challenge, and we don't know exactly what the relative abundance is of the intact protein to these cleaved domains. But what I can say is that if we integrate this AlphaFold, these prediction scores, we're getting a new view of something that might have some biological relevance. And so across these 6 proteins, each one we can see that there's progressive cleavage or single cleavage that removes the SH3 domain from the rest of these proteins. Now, further evidence that this is something that might be biologically interesting is the fact that these SH3 domains, even when they're separated, maintain their folded structure. And so these are structures generated with the ESN fold. There's some caveats, but trust me that I took the sequence from the proteoforms I identified there, and then predict these structures. And for the most part, the SH3 domains maintain the exact structure they had before. And so it's interesting to think that there could be some downstream effects or biological impacts from this process of enzymatic cleavage of a domain like this. And what's interesting, and kind of more supporting evidence towards this idea that this is something that it could be really going on, is that the first structure of the SH3 domain, and many SH3 domains that have been determined through X-ray crystallography since then, have often been isolated from the intact protein. So if you're not familiar with crystallography, often if the full protein that you express and you try to crystallize doesn't crystallize, or you can't get a structure, it's not uncommon to express just fragments of the protein and to see what can you get a structure out of that. And the SH3 domain was originally solved in that manner, and it's a very stable structure that, and I should mention this SH3 domain has many important functions in cell signaling, and it's so diverse. It's actually very hard for me to discuss in this talk alone. But I think it's a very interesting aspect of what we can do with TopPICR and the integration of AlphaFold. I think I see that I've gone quite a bit past my time. I should have been keeping an eye on that. So I'm going to skip the summary and get straight to the Q&A. But I really do need to acknowledge, of course, that this has been really great experience. I think that working with Vlad, Lily, and Moe on many of these different projects, PNL has been very insightful. And I hope that some of the ideas of TopPICR, you may find useful. I'm very open to working with others in collaborations and kind of implementing these ideas in broader context. So with that, I'll thank you and just leave the floor open for questions.
Thank you, James. That was wonderful. And I hope it brought all of the main points, at least for me, and advantages that this new tool might provide for people on this call. We are open for questions. We still have 10 minutes or so there is time to or raise your hand or just unmute yourself and ask the question. I think there is one question in the chat. Maybe we can start with that, James.
Yes, well, I have one question if I can ask now.
Sure, yeah.
Yes, I asked a question through the chat. James, you were explaining, I think, that part just at that moment, so I just read this question and then I listened to you. But even that, I couldn't understand very well. How can you assign or select a specific accession number in UniProt based on the molecular weight that you're observing of your proteoform? I mean, is it a step-by-step so that I can use, for example, for my research?
So you said based on the molecular weight.
Yes.
I think you're probably thinking about what I was talking about with parsimonious inference.
Yeah,
yes.
At the very beginning of your talk.
Yeah, the very beginning. Yes. Mm-hmm.
Yeah. So this is this idea of parsimonious inference, I encourage you to go look it up after. And so this is something that in bottom-up proteomics they do as well. What happens is you have shared peptides that can be assigned to different proteins. But what we often see is that some of these more computationally or entries in the Trimble database, for example, may not have as much evidence as one in SwissProt. And so usually there's also an imbalance between the abundance of the main protein and splice isoforms. So parsimonious inference is looking at the number of unique peptides or proteoforms that could be mapped to a particular gene and saying which one has the most and which one therefore provides the most evidence that for its existence. And so we take the peptides or proteoforms that are shared, and we lump them into that particular protein with the most unique identifications. That's what. So a razor proteoform is kind of like a razor peptide. And so razor is referring to Occam's razor. That we're making an assumption here about what is most likely to explain the origin of a peptide or proteoform. And so it's very much something that it really doesn't have an impact in top down like it does in bottom up. But I think it's important for at least kind of getting an idea like, well, what's most likely to explain where a proteoform is being derived from.
Okay. I see. All right. Thank you.
Any other questions for James? If there are no more questions, then I would like to thank you, James, again for a very inspiring talk. And I hope to see you all next time, mid-March. Thank you.
Thanks, Lily.